Buenos días a todos. Buena madrugada en Nueva York, obviamente. Agradecemos mucho que Estefania esté con nosotros a las 6 de la mañana. Pero bueno, estos son ganjes del directo, ¿no? Bueno, encantado de presentar a Fanny, que la conocemos más coloquialmente. No es una persona, digamos, desconocida para nosotros. Estefania hizo su tesis doctoral con nosotros, conjuntamente con la Universidad de Sevilla. Su tesis doctoral se centró sobre todo en filogenia, ecología y sistemática de anémonas en el océano Antártico, así que empezó en zonas polares frías. Fanny, cuando terminó su tesis, empezó su postdoc en la Ohio State University. Allí aprendió toda una serie de técnicas moleculares que han marcado su línea de investigación posterior. Es de las pocas personas en el campo de los nidarios que realmente conjuguen lo que sería la taxonomía clásica, con técnicas morfológicas, histológicas, estigmáticas con moleculares. Y su línea de investigación se ha centrado sobre todo en lo que sería filogenia, evolución de nidarios, de invertebrados, y las anémonas son su grupo por excelencia. Por su expertise prácticamente lo podíamos extender a todo el grupo de nidarios y un poquito más también a invertebrados, ¿no? Desde el lugar que está ahora, en el 2009 obtuvo una posición de tenure track en el Museo de Historia Natural de Nueva York y actualmente es full curator, que sería lo equivalente a cátedra en nuestro sistema, digamos, académico. Es la máxima responsable de colecciones y de las líneas de investigación de invertebrados marinos y me imagino que, aparte de sus campañas y sus trabajos, que son invidiables, no solamente en la Antártida, bueno, prácticamente ha visitado todos los océanos menos el Ártico, algún día llegará. Y la verdad es que, bueno, la charla de hoy ya la en inglés, las preguntas como queráis, pero de verdad que es un placer tenerla aquí a pesar del madrugón. Muchas gracias, Fanny, y adelante.
Bueno, pues hola, buenos días a todos. Espero que me oigáis bien. Oigáis bien. A ver cómo está mi voz por la mañana. Y bueno, como dice Josep María, estoy aquí un poquito para hablaros sobre la investigación que hago en el Museo de Historia Natural. Y bueno, como tenía toda, va a ser la charla en inglés, ¿vale? O lo hacemos así en un medio Spanglish, si hace falta. Bueno, pues ahí voy. So I'm a curator or the curator of marine invertebrates at the American Museum of Natural History. The museum is a private institution that has a mission, and that mission is focused in the research and the education and the outreach. They hold very large collections. We have about 24 millions of invertebrate specimens. And also in the museum, we have graduate school. The museum now has a high-level mentoring, and we can do PhDs now. And also in the museum, we do research. My research in the museum is focused on marine invertebrates. Within the marine invertebrates, my focal group, as Jose Maria said, are the sea anemones. And in my research, I try to integrate new tools. Some of my projects are just developing new tools that we need to be able to describe and to document the diversity of these animals and to try to find out how they evolved and how they got there. I also have a lot of, put a lot of effort in developing and promoting taxonomic expertise, because as we all know, in the last decades, there is a huge loss of experts and people don't know how to identify their animals anymore. So my research is specimen-based, I use specimens to test hypotheses in sea anemone evolution, and it's a collaborative research that is externally funded. So what are sea anemones? You probably know sea anemones are cnidarians, is the order Actiniaria. These are solitary hexacorals without skeleton. Okay, they live in every marine environment. And when I say every marine environment, that's what I mean. You can find them in the beach, but you can find them in the deepest trenches in this world. And they live in the ice, they live in hydrothermal vents, whatever you want, they are. As I said, they are cnidarians. Cnidarian is a group of very simple, relatively simple animals, about 10,000 species, very diverse group that includes jellyfish, corals, sea anemones, all these animals, and they are all together because they have nidae. Let's see if this comes out. Well, nidae are capsules that have tubules that usually have toxins, and all cnidarians have these capsules. All right, there are different types, and the one that all of them have are the nematodes. When I say that anemones are simple, but that doesn't make them poorly diverse. They are very diverse in every aspect of their lives, not only in their shapes, in their colors, but also in their biology. They reproduce in any way that you can think, sexually, asexually. They parental care, no parental care, budding, pedal laceration, dividing like this, dividing like that, everything. And same thing when they feed. They can be suspension feeders, they are opportunistic predators, but they can also be primary producers because they have the capacity to associate with symbiotic algae and other microorganisms. They are also able to engage symbiosis with many different animals. Here I put some of the more typical ones that everybody is familiar with, but they, as I said, they also associate with bacteria, and we just this year finally proved that they can also, for example, associate with chemiosynthetic bacteria. So very engaging in reproduction, in symbiosis and other interactions. So, as I said, anemones not only are awesome, but they are important. Ecologically, they form part of the suspension feeder community. In some cases, they have a structural role forming part of this symbiosis. They are also economic. They have economical relevance because they are beautiful, and a lot of people get them for their aquariums. And in some places, like in Spain, in the south, they eat them. And they are also a great potential for natural products. And some drugs against cancer are actually developed from them. So very beautiful, diverse, relevant animals. The systematics of this group is based in the internal anatomy and in these capsules, in this nidae that I was saying a second ago. And we look inside the form, the shape of the muscles, the arrangement of the mesenteries and things like that to differentiate these different species. Today, I'm going to talk a little bit about some parts of my research. My main focus, or the main focus of my research, is biodiversity and evolution. But later, I'm going to talk about a couple projects that have been funded in the last years that show you a little bit what can be done with these animals. the remarkable thing of these projects that I'm going to discuss today is that expanded my research program in the sense that, for example, I included new anemones for new areas, and we developed different new tools to try to study these anemones. So looking at the biodiversity of sea anemones, well, we have a very limited understanding of it. And I have been working since the last 20 years in describing anemones and trying to solve this issue of our poor knowledge of this group. So one of the things that I have been working a lot is in trying to make collaborations and trying to get the material or access to material so I can study them. And this is an example. In 2016, in collaboration with the British Antarctic Survey, I was invited to a cruise. We went to Antarctica, and from this cruise, but also from former cruises with the Paul and Stern and the Alfred Wegener Institute, we collected and we have access to different anemones that allow us to open our point of view of how the diversity and how these animals are. So here I'm gonna just talk a little bit about a recent description of specimens that actually represented a new family of sea anemones from Antarctica. Is the first family endemic from Antarctica, because as you might know, in Antarctica there is not too many endemic families usually, and it is a brooding sea anemone. All right, so here you have an image of several sections of the anemone, and in this top one I made a drawing so you can see that in the oral disc this anemone has evaginations, and that is where supposedly, because we didn't find offspring in there, they brood. So this strategy with brooding pockets in the oral disc has been only seen in the Arctic, in one genus in the Arctic so far. But another very interesting thing of this species is that actually when we found its phylogenetic position comes up here in this yellow superfamily, which basically is a polar superfamily, and is the first time that we found an anemone that is a burrower. Okay, anemones that burrow have a special morphology. They are convergent. They look like worms, more or less, to be able to burrow in the sediment. And there were so far no burrowers in this group. And the interesting thing about all these new lineages and all these new samples that we get is that they provide us with different insights in how these anemones are. And one recent one is that they show us that this hexamerous pattern that we have in our mind for hexacorallians, this is the name of the group, so it means that they are they have a symmetry of 6, okay, hexamerous. But looking in more detail, and because we are discovering all this diversity, we can think, or we are starting to think, that really that hexamerous pattern is not that prevalent in hexacorals. So we have exciting things to look into now. Once you actually get to know the diversity of a group, you get to know what species are there, that allows you, or allows us, to actually include this group of animals in in larger multidisciplinary studies. And this was the case, that I was in that cruise, and after 20 years I finally know my anemones, so we could identify them and include them in this study to try to enlarge this marine protected area in the South Orkneys Islands in in the Scotia Sea. In this study, we characterize all the fauna around the protected area, and what we found out is that camera systems are better than agassiz. There was a comparison in this study, and then the other main conclusion was that we really need to do detailed studies of the areas, because there is too much local variation, and just large geomorphometric patterns don't help. So before, I was telling you that anemones are very diverse, but they are simple. And one thing that we have been learning or finding out in the last years is that there is an amazing amount of morphological convergence. But not only anemones are convergent in relatively simple morphological characters, but we also find this convergence in very complicated behaviors. And I don't know if you know about this, but I love this example. That's what we have been looking into it lately, and is the evolution of the carcinoecium forming sea anemones. There is a group, or there are several species of anemones that live in symbiosis with hermit crabs. You all probably know that hermit crabs have a soft lower part of the body, and they need to live in the shell of a mollusk in order to be protected. So the problem that these crabs have is that once the shell is small, once they grow, the shell gets small, and they have to leave that shell and its protection to look for a bigger one. Well, anemones have kind of solved, or some anemones have solved this problem, and living together with the hermit crabs, what they do is that they produce a layer of chitin, all right, called carcinoation, that is made of chitin, I just said it, and they form, okay, a shelf that allows the crab to stay in the shell, because they basically make the shell growing at the same pace that the hermit crab does it. So basically, here you have an example of these shells that are made by the anemone, and the first time that they actually saw them, they described them as mollusk, because the shell was perfect, basically. So anyways, this is a very complicated, amazing relationship, and we have been looking in anemones, and we found that there are 4 different origins of this very complex behavior within anemones. So now that we know this, and we have a evolutionary framework to understand this, we are trying to see and look into why some anemones develop or whatever, develop or allow or can use this strategy while others don't. I have commented that there is a lot of morphological convergence, and because we have to look into the internally in the anatomies, we have to use histology and other techniques. And histology is a little bit time consuming, and also depends how your specimens are. It's difficult to use. So we are trying, in the last years, we have been trying to develop protocols to do micro-CT scanning in soft body invertebrates. Okay, so this basically, we get a 3D image of the anemone out internally, externally, and it allows us, hopefully, to see more characters and to restudy the ones that we use. Because these are soft body, it's not as straightforward as scanning a vertebrate, so we have to do some stainings, and we actually got some high contrast scans, and what we have been experiencing with this is that, yeah, we get scans, we get nice resolution, and this helps a lot to observe some of the characters, but we still don't get enough resolution to really see what we need for some groups. For example, these are all cross sections of different anemones, and this is, for example, one burrowing anemone, and these dots that you see here in the CT scanning are what we see here in histology. In this group in particularly, we need to count these processes, these muscle processes, so CT scanning is not enough yet for us, but it definitely helps. So right now, we have incorporated this technique, and whenever we do the description of a sea anemone, of something new, when we study something, as is the example of this new species from Brazil, we incorporate CT scanning. And like that, we can do renderings, see the anemone in 3Ds, we can look at the mesenteries down here and up here. As I said, it does help. So we have been working on that, and here in this work, what we were doing basically was making a comparison and a study of like, okay, can this CT scanning replace histology? It cannot. We still have to use the old techniques. Anyways, so more things that I have been doing. Well, another thing that this last project that I have been doing helped me with was to study the fauna of other places, because as Jose Maria said, I started in Antarctica and polar seas, but in the last years I have been looking into the diversity of tropical sea anemones, and to help in this task, I have been helping doing regional inventories and new records of fauna that was already known, but I have put a lot of effort in also in building capacity, in spreading the taxonomic expertise that so few people now have. This was through this project, we call it ART, and it was Advanced in Revisionary Taxonomy, and I don't know the S, what stands for right now. My mind, I can look it up later. Anyways, in this project, what we did, it was different groups, 6 groups of marine invertebrates, and we all got together and tried to make a set of tools and training to promote taxonomic expertise. So some of the tools that we did, it was an online glossary that is multilingual, it has several languages, where all the specific terms for the different groups are defined, they have translations, they have images, and hopefully the main specific terms are there described. We also made a set of videos in which it was a how-to videos. So we have all these where we explain, for example, there is a how to collect anemones, how to dissect an anemone, how to make media slides. So all these videos also came with bilingual protocols of how methodologies to study the different groups. And this project also had another part that was more direct instruction. And for that, we made some fieldwork courses where there was 2 weeks. We have international students from all over the place, but mainly from South America, actually. And in that course, as I said, it was hands-on. We had some theory at home in the classroom, but we also went and collected in the field and did histology, studied the anemones in there. Another thing that we did for this whole project was also a personalized training. One student, where is she here? This one, Jamali, actually came to the US, to the museum, and she stayed with me and in my lab for 8 months with the aim of give her a full training on sea anemones. And actually, she just got accepted in CUNY University, so she's going to be doing her PhD in a partner program between the school at the museum and CUNY University. And the last project that I want to talk to you about today is another example of collaborative research, and an example where we did develop tools and we use them for address bigger questions, I guess, questions that other people than me might be interested on. So the objective of this project was to construct a robust fossil calibrated phylogenetic tree using genomics, using ultra conserved elements for the group Anthozoa. This is the class within cnidarians. The Anthozoans are the ones that only have polyp. They don't have jellyfish in the life, and it's the group that includes. Well, I'm gonna say that later. Anyways, what we wanted to do with this project was to construct this phylogeny to try to solve the relationship among the different groups of Anthozoans, and to use this tree to understand the evolutionary gain and loss of some of the morphological key characters that this group has, okay, in order to see how adaptive this group is and to look through the past to see how we can inform the present. I'm going to explain myself, I promise. So, as I said, this is Anthozoa, the group that includes all cnidarians with only a polyp phase, includes the corals, the anemones, the sea fans, the gorgonians, all this group of animals. And as you know, they are one of the most important structural groups in the sea, not only in the shallow waters where we have all the coral reefs, but also in the deep sea where octocorals and other anthozoans make all the structures for all the rest of the animals. So this group, even though it's there, is important, we really don't have a clue of many of them, of the relationship within these animals. We don't know how they are related to each other, and this is mainly because molecularly, this group is a little bit special, probably because it's so old. Okay, the mitochondrial genome evolution is very slow, so it's not that easy to find molecular markers that are different enough. So what we wanted to do is to develop new tools to be able to address their relationships. And we did that with available genomes and transcriptomes. We designed some baits to capture UCEs and other parts of the genome. UCEs, I don't know if you know, they are sections in the genome that are very conserved across the whole tree of life, basically. And a lot of research lately has been using those regions to locate them and then to try to sequence the flanking regions where the sequences are more variable and you can compare the different relationships among animals at different levels. So those were not developed for anthozoans, and that's what we did. And through a lot of molecular and bioinformatics work, we developed this set and we were able to construct this phylogeny that we calibrated with fossils. And like that, we finally got a phylogenetic tree where all the different relationships of the different groups within Anthozoan were finally resolved, or that's what we thought. At least we have support for the different relationships, and also we have dates. And what did we do with this? Well, this is the interesting part. Once you have a phylogenetic base that you can actually base your work on, you can find out things like when and how, when more or less, when is that these different types of corals or of anthozoans develop their skeletons, and was that in specific conditions of the ocean. And what we found out is that yes, actually, when we look into diversification rates and into when the different types of skeletons that this group shows appear in the evolutionary history, we saw that first, that basically the paleoclimate had shaped the evolution of their skeletons and other traits. All right. And the first thing that we found out is that anthozoans are way older than we actually thought they were. And now we have estimations of them appearing about 800 million years ago. Okay, that's 250 million years ago before the first fossils that we have from them appear. And all the things that we discovered or that we found out is that the ancestral polyp was actually a solitary polyp that had bilateral symmetry and that it didn't have skeleton. All right, and then we found out that colonial growth and the ability to precipitate calcium carbonate, which allows them to make all these nice skeletons that all other animals used to live on them, appear the coloniality in the Ediacaran almost 600 million years ago, and the ability of creating a skeleton in the Cambrian. But this tree, what it's telling us to, is that first we have, and this is related with the previous slide, is that what we had is in the Cambrian, we have a solitary polyp that actually creates a skeleton, but this happens when the ocean conditions were favoring calcitic conditions. So this means that the skeleton that the anthozoans could make at that point it had less content of magnesium. All right, and that is the skeleton that characterize here all the octocorals. All right, all the gorgonians, all the soft corals, all those. And that is a different skeleton that later on, way later on, here in the, I think 380 million years ago, the scleractinian corals evolved and made those skeletons that they have now, which are made of aragonite, right? So the point of this is also that we can see, I mean, here you probably cannot see the lines, but we map the different mass extinctions during history and coral reefs and ocean acidification events along the evolutionary history of Earth. And we saw that, again, these corals, for example, anemones, were diversified after a coral crisis. So when the conditions were not optimal for corals to create their skeletons, their aragonitic skeletons, anemones suddenly occupied all those areas that they were occupied. So what this study tells us is, first of all, that anthozoans have been there for many, many millions of years, all right, that they have survived all massive extinctions that we know of, and that they change and they adapt. And if we continue based on their story, if the condition continue to be warm and acidic, scleractinians and the coral reefs, as we know, are going to have a very hard time. But other groups of anthozoans are going to replace those, and in a different way, anthozoans will still be with us, probably different ecosystems, but still they will be there. Okay, very quickly. So I have been telling you that Anthozoa, that we developed these molecular tools, these UCEs, in order to try to get support to study these animals. And this complements another area that I have been looking lately. And this is the study of species delimitation and cryptic diversity that we found in a group of anemones, the ones that host clownfish. As you might know, I mean, this everybody knows, come on, Nemo. So anemones are in symbiosis with clownfish. They live among their tentacles, and this was a very successful symbiosis. And once it came, actually it was great for the fish. They diversified like crazy with 30 species, and we know all about the fish. 12 million years ago, they diversified in a single radiation, blah, 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 a lot of knowledge about the fish. But nobody happens to look into the host of this symbiosis. And so what we find is that there is an unpaired diversification in the host and in the symbiont relationship here. We have a lot of species of fish, and we only have 10 species of anemones that host them. This seems a little bit strange, so we wanted to look into what was the reality of this, and I did this with a former postdoc of me. He was at the museum, then Titus, then he went to Switzerland to actually study more genome level research, and now he's in Alabama. So what we did, and the problem is pretty clear, all these images here correspond to the same clownfish anemone, Entacmaea quadricolor, or the bubble tip anemone. As you can see, they are very different, and also the geographic range of this anemone goes from Eastern Africa to basically Australia, which is a little bit difficult to think that it's all the same thing. So in the last years, we have been studying this group, and first of all, we studied it with the 5 mitochondrial and rDNA markers, the ones that we usually have been using for anemone systematics for the last 20 years. And just with those markers, and don't really provide much resolution, what we saw is that there are at least 3 independent origins for this symbiosis within the anemones. So different anemones from different families have evolved this symbiosis. So this means that we have a huge taxonomic revision in front of us, of course. And when we looked into markers with more capability to look at relationships at lower level, we also see that there is way more molecular diversity than we thought. So here basically we have 1, 2, 3, 4, 5, 6, 7 different species of the bubble tea anemone, just based on the rat-seq data. Since we found all this diversity, this prompts us to write a proposal and a grant to actually look into this group in detail. And now we are doing a holistic approach, looking into everything and microbiome, the anatomy, the molecular part of it, with regular markers, with the species delimitation markers. You see everything that we can basically to try to solve this mystery. And I'm gonna leave you, so I don't get you too tired with anemones, with one other part of my job, which is popularizing science. As you probably know and all do, is that we have to be able to convey and to explain to the general public what we find with our research. And in the last year, and being at the museum, I have been able to do this and to participate in a lot of different venues and different programs to try to popularize the science that we do. So I have been doing blogs of research and work in the collections that we do, or we have talks or blogs, also little magazines for kids. But I wanted to bring this up because one example of one thing that we do, it was very inspiring for me. We had this behind the tour scene for a group of preschoolers. We are talking about 3 or 4 years old kids from one school in New York. They came and visit the museum, and also they visit the collections. So my assistant and I show them everything, and here they prepare a booklet of what they have learned, and with pictures and everything. And this was already pretty sweet, I would say, with all the pictures. And if you get to read some of the comments of the things here, they are really funny. And they also sent us this, which kind of got into my heart, very deep. Dear Estefania and Lily is my assistant. Thank you, the force. This is the school. But they didn't stop there. They actually create these books and these drawings and these models, and they did actually an exhibit in their school. They make this exhibit with all the books, the pictures, the drawings, some models that they do. And they made an exhibit in their school, and actually people went and see the exhibit, and they actually collected money, okay, not a lot, but some money, which they donated to take care of the collections, which I actually found, as I said, that it was very, very inspiring, that, well, at least they learn something, at least they know what marine invertebrates are, and maybe perhaps I inspire one of those to keep on this. And because I don't want to tire you too much, this is all that I have for you today. I realized that I forgot my thank you slides to all the funding sources, the National Science Foundation, and all the people that have helped me along the way. You know, many people at the museum, former members of the lab, collaborations, and thank you for listening to me. I'm happy to ask any questions, to respond to them. That's it.
Fantástico, Fanny. Muchísimas gracias por la conferencia. Evidentemente que súper interesante, especialmente para los que estamos más cerca de este tipo de investigación. Bueno, tengo dos... Bueno, se me ocurren un montón de cosas, pero yo creo que hay que ser moderados y sobre todo tú que ahora estás casi a las 7 de la mañana.
Estoy despierta.
Siempre tenía la idea de que todo este proceso evolutivo de los organismos tan simples como las anémonas o los octobarrelarios vienen de hace, como has dicho muy bien, más de 600 millones de años. En esta larga carrera en la historia de la vida del océano en el planeta, ¿Conoces algún grupo que realmente se haya extinguido? ¿O son básicamente un proceso que, gracias a la simplicidad o a la forma o su adaptabilidad, realmente con pocos cambios aún han persistido y, como has dicho, han superado todas las crisis de, digamos, de extinción?
Hombre, hay un grupo que está extinguido, son los corales, los tabulates, no sé cómo se dice esto en español, qué desastre, los tabulados, ¿no? Los corales tabulados, que son los primeros corales que tenemos fósiles y estos también, estos están listos. Los corales que tenemos ahora, las escleractinias de ahora, no vienen de ahí, se supone. Tienen un diferente, tienen un origen diferente. Entonces, sí, estos, ¿vale? Los tabulados se extinguieron, pero bueno, de alguna manera las escleractinias agarraron el mismo tipo de esqueleto.
O sea, que ha habido pocas víctimas de este grupo a lo largo de las extinciones.
Bueno, y otro grupo que nosotros, que es este Relicanthus, es que no lo he comentado, es que hay tantas cosas que comentar que no se puede. Porque en el 2014 descubrimos esta anémona gigante que se llama Relicanthus, que vive en hydrothermal vents, que tiene como 1 metro de diámetro y 2 metros de tentáculos, es gigante. Pues pensábamos que era una anémona, tal, no va, y ahora resulta que no es una anémona, no sale con el orden Actiniaria, y todavía no sabemos si es un orden diferente que se ha extinguido y es el único representante, si es una anémona, que eso, lo mismo, ¿no? Que solo queda un representante. Todavía no sabemos
Fantástico.
mucho.
Bueno, hay una pregunta aquí en el chat, como verás, ¿no? Bueno, aparte que te agradecen muchísimo la presentación. Podrían ser, digamos, indicadoras de esas condiciones físico-químicas, al menos de las masas de agua cerca del fondo, o tienen una gran adaptabilidad.
Pues hay de todo, como en la viña del Señor. Hay algunas que son muy sensibles y hay algunas que son más robustas. En general, hay muchos estudios, sobre todo de simbiosis y de coral bleaching y todo esto, que se hacen con anémonas, porque las anémonas son mucho más resistentes que los corales, ¿vale? Pero el problema que tienen las anémonas es que muy poca gente las estudia, y aunque son perfectas para estudios de calidad de agua y de todo, pues no las estudian porque no sé si son difíciles de identificar y las ignoran. ¿Sabes? Pero
No,
sí, tienen un gran potencial para todo tipo de estudios.
bueno, yo puedo certificar que realmente no son nada fáciles. Lo que pasa es que luego de ver la última parte que has explicado, sobre todo la parte de formar nuevas generaciones, yo creo que, digamos, puede ayudar a que realmente se estudie, ¿no? Creo que una de las cosas que me impresiona es realmente que si se enseñan bien con cursos como los que has explicado, ¿no? En un ámbito como el museo, los caracteres básicos de muchas especies, realmente se acaban conociendo y se acaban identificando. Yo luego a veces pienso que quizás con excepciones, ¿no? Realmente no salimos de la universidad o, digamos, suficientemente formados para poder identificar incluso los grupos, ¿no? En ese sentido, yo creo que chapó por esa capacidad de hacer cursos, de animar a la gente y tener este aprendizaje, ¿no? Esto lo veo muy claro, ¿no? Otra cosa, bueno, ya iremos acabando, que es muy tarde, sobre todo para ti, pero hay una curiosidad. Últimamente hemos leído que tanto en corales como en octocorales, corales, digamos, hexacoralarios, se empiezan a conocer casos de lo que seríamos de hermafroditismo. ¿Hay algún registro que en anémonas realmente puede haber especies que sean hermafroditas?
A mí, cuando he dicho al principio que las anémonas son diversas en todas sus facetas, sí, hermafroditas, pues no hay muchísimas, pero hay, no te voy a poder dar un número ahora mismo, pero bueno, hay bastantes casos. Y uno, por ejemplo, es el de la Epiactis georgiana, de la anémona esta de la Antártida, que cuida la, la progenie,
Sí.
¿vale? Sí, en corales también y en octocorales. En general, todos los anthozoos son muy plásticos y tienen un montón de, ¿sabes? Todo tipo de revolución, de estrategias reproductivas. Las anémonas son geniales. Hay algunas que un tentáculo se lo comen y ahora de ese tentáculo sale una anemonita nueva, ¿no? De un trocito de la base del discopedio, si lo dejan por detrás, sale una anémona nueva. Hacen partenogénesis, hacen, ya te digo, lo que sea.
Tenemos una pregunta aquí que dice, ¿cuál es el marcador molecular más fácil para identificar las especies?
Pues la respuesta triste es que no hay, ¿vale? Nosotros lo que hemos estado haciendo últimamente, o sea, en los últimos años, es usamos ADN ribosómico, ¿vale? El 18S y el 28S, marcadores nucleares, y luego mitocondriales, el 12S, el 16S y citocromosidasa 3, ¿vale? Y cogemos todos esos marcadores, los ponemos todos juntos y si tenemos suerte nos distinguen a nivel de género. Para anémonas y para anthozoos en general, COA1, la citocromosidasa 1, no funciona porque no hay variabilidad suficiente. Dije antes que el genoma mitocondrial evoluciona muy despacio, ¿vale? Hay mucha convergencia ahí y entonces no varía. Entonces, pues tenemos secuencias de dos géneros distintos que se diferencian en una base solo y cosas así. Entonces, por eso es por lo que hemos entrado en genómica y hemos intentado desarrollar estos ultra conserved elements porque es que no tenemos marcadores. No hay una cosa mágica. Ojalá.
Ahora me acaban de hacer una pregunta aquí presencial. ¿Conoces, como dice que hay una anémona que vive adherida al hielo marino antártico?
Sí,
¿Te
sí.
suena?
Sí, esta la describió, es una de estas anémonas que se entierra, pero en vez de enterrarse en el sustrato blando, se entierra en el hielo y vive ahí boca abajo. Es una de las fotos, la tengo aquí, a ver si, lo que pasa es que si hago esto igual se me vuelve loco, ¿no? Esto, al principio, sí vive ahí colgando del hielo. Es que estos bichos son muy fuertes. A ver, llegaré al principio. Sí, esta la describió mi amiga, mi colaboradora, Meg Daly. A ver, váyatela. Aquí. Sí que he hablado, ¿no? He puesto fotos.
No, las fotos son geniales, la verdad.
Es que estos son muy bonitos estos. Aquí. Esta de aquí es una foto de la plataforma del hielo antártico y estas cositas blancas colgando son las anémonas.
¿Y esas comen krill?
Pues comen lo que haya por ahí, lo que agarren.
Fantástico. Bueno, a ver, yo ya la última pregunta es si es una opinión. Has hablado bastante de que ahí tenemos una crisis de expertise en lo que es la identificación de especies. Evidentemente que el futuro pasa por congeniar la taxonomía morfológica que se le dice tradicional, pero ha sido la de siempre, morfológica y histológica con la molecular, ¿no? ¿Cómo ves el futuro de esta investigación? Porque, claro, somos muy conscientes y nos preocupa muchísimo lo que es cómo va a evolucionar, cuál va a ser la pérdida de diversidad realmente en los últimos años y los próximos años debido al efecto antropogénico, pero realmente si no sabemos lo que hay, no sabemos identificar lo que queda, ¿cómo lo podremos saber? Digamos que cómo es desde el museo, desde el museo siempre lo vemos como un lugar de referencia a nivel mundial de lo que es la preservación de la biodiversidad y del conocimiento. Desde tú estás en ese, ¿cómo lo ves? Con tus jefes, con la gente, tus colaboradores, con los proyectos en Estados Unidos, ¿cómo ves el futuro de que realmente no se pierda esa capacidad de identificación?
Pues está complicado porque hay como intereses conflictivos, ¿sabes? Por un lado, toda la comunidad científica está súper, que se me está viniendo el inglés, perdón, está muy al loro, very aware, están sobre este problema, ¿no? De que no hay taxónomos, no hay científicos que sean capaces de identificar los organismos. Pero claro, el problema que tiene esto es que al final, y a mí me pasa, ¿no? Cuando yo llego y tengo un estudiante, de repente no quiero que haga algunos, o que hay diferentes proyectos, pero tú tienes que pensar que un estudiante, cuando sale con un doctorado, tiene que tener 3, 4 papers publicados, tienen que estar en revistas con un impact factor decente, porque si no, pues no va a encontrar trabajo. Entonces ahí entras en un gran conflicto, ¿no? Porque la investigación tiene que ser sexy, tiene que venderse para que tu estudiante pueda vivir y comer y seguir haciendo investigación. Y eso no vale muy bien para el trabajo taxonómico, que te pegas 5 años y sacas un artículo que es una revisión así, eso sí, pero ¿qué? Que va a zootaxa con un 0.8 de impact factor. Y ahora llega alguien, hace un trabajo molecular con UCEs, directo, Nature. Entonces está complicado porque no, hay como dos fuerzas, se quiere mantener, pero se quiere seguir haciendo cosas que vendan rápido. Entonces, no sé, yo estoy un poco preocupada en realidad. Pero también tengo que decir que hay un montón de esfuerzos en general para eso, para desarrollar nuevas herramientas, website, citizen science, ¿sabes? Estamos trabajando en ello también, poco a poco.
Genial. Pues si os parece bien, muchísimas gracias, de verdad, por estar con nosotros de ese madrugón que te hemos hecho hacer, ¿no? Fantástico, por así te hemos conocido un poco más. Y Fanny, para los amigos, pues te dejamos descansar y sobre todo hasta muy pronto.
Vale, pues muchísimas gracias. La verdad que gracias porque después de tanto tiempo en colaboración con vosotros y que mi tesis básicamente vino de allí, es la primera vez que he dado una charla en el instituto.
Bueno, a ver si sentamos cátedra y conseguimos que algunos más también lo hagan.
Vale.
Bueno, hasta muy pronto.
Vale, si alguien quiere más detalles, por supuesto, me escribís y no, os puedo machacar con anémonas.
Aquí algunos de tus compañeros te dan, algunos que conociste en campañas también, ¿se acuerdan? Y bueno, y también hay un comentario que seguiremos más en contacto.
Perfecto.
Fanny, si quieres puedes echar un